Thank you so much for inviting me back at Science and Beers. I hope I did a good job the first time around, and apparently I did. So I'm so happy to see all the people here, all of you guys here today. It means a lot. It's a great end of I've been around talking a lot because it's been the Forskningsdøgn a couple of weeks ago, and I did a lot of talks that day, and this kind of marks the end of that. So I'm warmed up. I hope you have been warmed up because we have just seen Gecko, and I actually just wanted to ask Gecko a thing because I imagine the first time you went on stage, how did you feel?
Probably a bit anxious.
Yes. I actually know it from myself. I actually played vinyl records, but I've been on big stages and tried to perform for people. So I know that feeling of when you have to go up the first time, but then you get over it as you do it more and more. I want to tell you a different story, and now I can see that some of you are old enough to have been on a ski journey once in your life, or twice maybe, and some of you may be old enough, as old as me, to have actually gone through that era, and then stopped because you had kids, and then went for a long time, and then suddenly you had to go on a ski journey again, or travel with your kids. And this is my son here, and we went out skiing this year for the first time in I've felt like ages, 12 years I think, from the last time I was on ski. So this was his first time. He's 5. He's all right. He's good now. So you know. But on the first day, he goes into the skiing classes. He learns how to ski down a very flat hill. And then he tells me that, Dad, I'm all right. I can go with you up on the mountain today. I'm like, maybe not. But when he says this, I know I can't argue with him. He has to go. So up we go, both of us to the top of the hill. And the first hill, or the first ski hill, you have to go down from here, and then it's smooth sailing all the way. But the first part is a really steep part. And I'm really not a good skier at this point. I haven't been skiing for 12 years. And my kid is between my knees because that's the only way you can keep them like close when you are trying to learn how to ski. And then we go roughly 2 meters out this hill, and then I just get the biggest panic attack in my life. And I imagine him just going off the hill down into the valley below, and we're standing, my legs are shaking, my hands are numb, and I'm shaking, and my head is just whoa, sweating, running down my spine. But we made it to the bottom of this hill, right? But it just means that everybody probably has had this feeling once in their lifetime. Maybe it's getting up on stage, or maybe it's going down a ski hill with your kid through your knees. And I'll get back to this story in a little bit, because later that week we got better and better. But today we're going through a couple of stages in order to try to tell you a story. I'm going to tell you a little bit about who I am, because I think it's important that you all know a little bit about my background. And then I have 2 science papers, or scientific papers, that I'm going to go into detail with. And the first one is relatively easy. The next one is pretty heavy. But I'm going to cut them out into easy pieces. And then after the second one, we're going to have a break, a beer, and then we're going to go into new ways to treat anxiety and PTSD. And then in the end, we're going to have a lot of questions and talk about what's up. Sounds good? Good. All right. So this is me, or this is how I see myself, because I'm an engineer by training, and I call myself a neuroengineer. Meaning that I'm definitely not a medical doctor, and I'm definitely not a psychologist. So if you try and have questions about these, then I know a lot about neuroscience, but I'm not here to treat anybody, and I'm not here to try and figure out how your mind is. If you think it's exciting what I'm telling you about and want to know more, I'm at LinkedIn a lot. I try to post a lot of posts in there. I'm speaking about psychedelic drugs and psychiatric disorders. I'm also on Twitter, not so much though, but find me if you wanna follow up. So my lab is trying to understand psychiatric disorders from the core of the disorders. And when you look at mental health and mental health disorders, you can kind of put them into two groups. So there is disorders that are driven by genes and genetic. That could be Fragile X syndrome, which is a almost 100% genetic disease. And then there's disorders that are more what we call maladaptive. So it's things that we learn, and it's the brain that mismatches things and does things. So these are the things that I'm interested in. This is where we learn stuff that is not nice, but we can also unlearn these things again, right? So this paper actually came out not so long ago. I think it was in January or December. And I brought it because it tells you something about the complexity of our brain. So these scientists, they looked at the fruit fly and they counted all the nerve cells inside the brain of the fruit fly. And the fruit fly has 3,016 nerve cells in their brain. And every time one of these nerve cells are connected to each other, so they touch each other in ways to communicate, they go like this, they hang up, and every time they do that, it's called a synapse. So that's a place where they touch each other. And I may say synapse and you say what? And then just try and remember, oh yeah, but that was when two neurons are touching each other. Connections, I will also use that word. But they have, from the 3,000 nerve cells, they have over 500,000 of these, 500,000 of these connections between cells, right? So if we try and multiply this and make it into a human brain, then we have millions of neurons and probably billions of these connections between neurons. And we are going to talk about very few of them. So just try and remember that. This is a complex organ that we have on top of our head. So this is this is how one synapse, one connection between two neurons look like. It's just a drawing, but we are becoming really good at looking into the brain and looking at what is happening at these connections between two nerve cells. And we can do that by putting electrodes or light cables inside the brain of mice or rats, humans as well. But we have a light cable going out, and then we can actually look into the brain of the mouse, specific areas, and then we can look at neurons in the brain. So I want you to remember these things, so you can forget the rest, but just remember some of these things when you go home. That anxiety has a clear neurobiological cause. There are neurons or nerve cells in the brain that causes anxiety. And you can learn to fear, but you can also unlearn it again. It does take time and it does require psychological work. But you can get help, either psychologist or, as we're going to speak about, there's also new medical treatments that may be able to help. If you are, as me, older, then you might need it. All right, but we're going to go back in time a little bit. This guy is a professor in London. He's called John O'Keefe, and he was one of the first persons to put an electrode down into the brain of a rat. And you can see it's old because this is a very pixelated video, but you can also see that this is a rat and there's a cable coming out of it. And he put an electrode down into the hippocampus of this rat. And the hippocampus is an area that is involved in learning and memory. And let's hope we get the sound. Yeah, great. So what you're seeing here is actually they put this cable into the mouse brain, and then they picked out different nerve cells to follow over time. And the one that we're hearing is the green one up here. And you notice that when this rat is running around in this big open square, that particular cell is only firing and only making signals when the rat goes into that area. So that's it. So these cells that John O'Keefe found are called place preference cells. And they only fire when a rat is in a particular space. So if I go to the back of the room, my cells would fire. One of the cells would fire, and when I'm up here, another cell would fire. So when I move around in the room, different cells will fire in different places.
Is that done after you found out? Like, is that ellipse drawn afterwards?
No, that's it's drawn afterwards. But I made one as well. So, but yeah, in the brain there's different nerve cells. There's also different nerve cells that tells you about speed and motion and stuff like this. But then last year, these people, they didn't look in the hippocampus, but they put electrodes down in another area in the brain that's called the amygdala. And the amygdala is an area of the brain that is involved in fear. It's also involved in interpreting feelings and sensory input from the surroundings. It's something that's involved in sex drive and going out, and also these bodily function feelings that connect to our brain. So they took electrodes, put them down into the amygdala, and then they had the mouse sitting either in the home cage, or they took the mouse and then they lifted up the mouse very high, right? And what happened then? They found cells inside the amygdala that fired. So the blue lines up here, that is the same as the noise you heard before. So once they lift up the mouse, these cells start to communicate with one another. They start to send signals to one another. And at the same time, the heart rate went up on the mouse. So they could actually lift it up, and they found cells that reacted to this being lifted up, being up high, and being probably afraid of the heights. So they went on a little bit because, okay, we can lift it up. Maybe it's also just the hand movement, or maybe it's something else that does that these nerve cells are firing. But they made another experiment where they put the mouse into a new cage, and one of the cages had walls that were made of see-through plexiglass, and the other one was just an open high space. And then they saw that the neurons or nerve cells in the same spot were firing more the more exposed the mouse was to the height, to the danger of the height. Okay, so there was nerve cells in the brain that responded to fear of heights, but they went on and tried to stimulate the mouse in different ways. So they also gave it a loud noise and found cells that reacted to the loud noise, cells that reacted to looming objects, cat odor, and of course high places as we looked at before. So that means that inside the amygdala you have cells that react to different kind of dangerous stimuli from the world. So it's completely normal to get this response. It's completely normal to fear. This is something our brain does. It's made to fear. It goes all the way from the mouse to the human, and it's conserved up into through evolution. So it's a really important survival technique. I know this one is in Danish, but it's a Danish artist that tries to inscribe how does it feel to have anxiety when you are a human. Because a human is not a mouse, and I know that if you have anxiety and feel anxious as a human, you have very different ways of feeling it. And she's, you can find her here. She makes books about ghosts that have anxiety and makes a lot of nice photos of it. But the important thing to state is that even though it's different from person to person, it's also different from a mouse to a human, right? And what I'm talking about today is mainly things that have been done in mice and rats. But we can translate, because the mouse and the rat has an amygdala, it has an hippocampus, it has the same brain areas as we do, and we can translate it to humans in some form. But just remember, it is a mouse. So I want to mention two things to you that is really good when you do if you have anxiety. So there's two things that I want to tell you that it's really important to know, because this is things that can help you in your daily life. And one of these are breath work. So if you can do stuff with your breathing that can lower your anxiety, you can actually immediately lower your anxiety. So if this is before you have to go to an exam, before you have to get up stage and talk to 100 people, you can make short inhales followed by long exhales. And that's really important because there's a ton of ways that these kinds of breath works are explained. But the important part to remember is that you have to have a short inhale and then a long exhale, right? And then you have to repeat it a couple of times. So it goes like this. Maybe you want to try it one more time with me? Huh? That's not too important. The important part is the quick inhale and the long exhale. This one I'm showing up here is called the 4-7-8 breathing. There's also something called box breathing. And if you are a kid, you might be told to blow out candles on a birthday cake. So there's very many different ways to do this, but the important part is the quick inhale and the long exhale, right? This will lower your anxiety.
So what's the difference with the holding part?
It helps to get more oxygen into your blood. So if you have filled your lungs and you hold it, then you get more oxygen in your blood. The other thing that's super important, that's to have a good night's sleep before you have to go and do something you know might trigger an anxiety response. And this is my sleep from April 5th. My wife is a sleep researcher, so she makes me wear one of these. And she also told me a bit about sleep. But you can see, I don't know how many of you have a Fitbit, but it probably looks the same if you have something else that tracks your sleep. So you can see you go through different stages throughout the night. That's called deep, light, REM, and awake. And in the beginning, we sleep very well. So we are actually in deep sleep in the beginning of our sleep phases. And this is when the body restores, and you can actually feel well even if you only had 4 hours of sleep. You can be okay. But in the second part of the sleep, that's when you have what is called the REM sleep. And if you had kids like me and you've been up all night, been waking up many times, you also know that you feel a little bit mentally tired the next day, even though you had a lot of sleep. So the REM sleep is something that comes after many hours, and it's about restoring your mental health. So while the body restoration is for your physical health, this is for your mental health. So it's really important to get enough sleep before something. Okay, so we'll talk a little bit about something different. That was the 2 points I wanted to make. I don't know if you know this guy. He's called Ivan Pavlov, and he's a Russian. He's a Russian psychologist from back in 19-ish, and he made this experiment where he was breeding dogs, so he had a lot of dogs with him, and he noticed that when he was trying to feed them and they were eating, they all started to drool. But since he had a lot of dogs, he wanted to get them all together at once, so he didn't have to go around feed them individually. So he started to ringing a bell, and all the dogs came running, and then they ate at the same time. But he noticed that after a while, when he was ringing this bell, the dogs started to drool before he fed them. So this is called conditioning. And it's named after the paper that Ivan Pavlov wrote back in 1900 something, and he got the Nobel Prize for this in 1904. But it's actually a really important experiment for anxiety and fear research, because we can do the same thing with a fear response in these mice. So we can take the mice we had before that had an electrode planted in their head, And then we can play a sound, and at the same time we can trigger a small electric shock on the floor that it's moving on. And when a mouse feels electric shocks on its feet, it stops and freezes. So that's what we call a fear response. Just stops and freezes. Okay? They're not hurt, it just tickles a bit. So these Korean researchers, they wanted to study this phenomenon, and they wanted to learn how is this happening, how is these things, how is the sound coded with the electric shock. So they did the same thing as before. They had the mouse, and this time they put an optic fiber into the brain, and not only in one place. They actually placed 3 optic fibers, one in the hippocampus, one in the prefrontal cortex, and one in the amygdala. And then they did something that was really smart. They actually injected a dye into the mouse that once these neurons started, or nerve cells started to talk to each other, they were colored red, so you could find them. Then you could look into the brain and you can find the cells that were lighting up, and because they were colored red. So they're looking into 3 regions of the brain, and then they're exposing the mouse to the electric foot shock. And when they do this, the mouse gets a fear response and it freezes. And then they could find specific cells in the amygdala, in the hippocampus, and in the prefrontal cortex that all lit up and were activated when the mouse had this response. When they only played the sound, the mouse doesn't freeze and they don't see the same thing. So then they started to play the sound and the electric shock at the same time. And they tried to follow what is going on with these cells when we do this experiment. Because they could see the cells and they could follow them in real time once they were doing the coding of these 2 sounds or stimuli together. And what they noticed was that the cells in the prefrontal cortex, so that's the frontal part of your head, they started talking together, talking more together. So they got more connections, they made more synapses, and they had more connections between them after you have coded the electric shock with the sound. The cells in the amygdala didn't do this, and the cells in the hippocampus didn't do this. They just went back to normal. So this coding of these 2 stimuli, it was saved inside the frontal part of the brain. And you could see that when you played the sound and the electric foot shock, both of these responses resulted in a freezing response in the mouse. So it was standing still and it was afraid. So this dye was actually pretty special because you could, once you had this dye in the nerve cells, you would actually light on them with a green laser, and then you could turn them on. So this is like a mouse that you can steer around like you have a car or something like that, right? I don't know the English word of a car that's remote controlled car also. Yeah, cool. So, but what do you think happened once they lighted green light on these neurons? Yes. So the mouse, these cells get activated, the cells in the amygdala get activated, and then the mouse freezes. So this is before, off, and then on the laser, and then after the laser. So you can see as soon as you turn the laser on, there's nothing happening, there's no electric shock, there's no light, the mice freezes up. So that means that the fear response, the coding of the sound with the response is saved inside the brain of the mouse, up in the front part of the brain. And you can, if you want to translate this into humans, it's a little bit like post-traumatic stress disorder, where you get a stimuli and some frightening trauma coded together. They don't have anything to do with each other. They could be the smell of fresh baked bread coded together with a car bomb explosion in Afghanistan. And then suddenly when you walk down the street and you smell bread, you get the fear response from the bombing. And that's actually saved inside the brain. So the good thing about it is that you can make this go away, but it takes some time. After this, then the scientists, they just played the sound for the mouse without giving it the electric shock. And when you do this enough, the mouse realizes, okay, the sound isn't as dangerous as I thought it was. And then the sound, or the response to the sound, disappears over time. And they followed the cells in the frontal cortex, or in front of your brain, and they could see that the connections that was formed between the cells when you were coding these two stimuli, they started to disappear. So they were disappearing again, and the memory of the sound and the stimuli was no longer. And after stimulation, the sound didn't induce this fear response, this freezing response in the mice. So this is similar to what you do if you want to treat post-traumatic stress disorder or anxiety. And if any of you have been at a psychologist and tried treatment, you would know, you would be told about getting up a ladder and getting a bit up to face your fear. And this is what I try to illustrate it about. You go up the ladder to a certain point and then you use breath work to calm yourself down. And then over time, this exposure to the fear response will also lower your fears. And this works very well if you have fear of heights or fear of spiders. I know that if you are a human being, like the rest of us, this can be really complex. So the anxiety in a human can be super complex, but it's still the same idea. In humans, you may have to tease it out. So you may have to take, many times you can have many anxieties piled on top of each other and you don't really know what is what, and you have anxiety for the anxiety and it just keeps on going. But you have to take it out and do one at a time. You can't just say, today I'm gonna go out by myself and hopefully I won't get an anxiety response when I'm out in the town. No. Do one little step at a time, preferably with a psychologist, right? But then you can also get better. That's what happened to me and my kid. We took this every day for 5 days. And even though I didn't get much better, I was still horrible at the last day. I didn't get the same big fear response when we were going down the ski hill on the next days. So as soon as I knew that we could do it, even though we were falling and tumbling around, it got better and better and better. And on the last day, I didn't fear going down that one. All right. So I have a video to play for you, but I'm not gonna do it before they. I'm gonna give you a little bit heads up because then they can cut it out. All right. So it's a teaser, yeah. So you may know it. It's from The Office, and it's a very famous clip about fear conditioning. I think this is a really good time to take a little break, and then when you come back, we'll talk about different treatments and how the treatment will work on the conditioning of two sounds and stimuli, and we'll talk about MDMA, and we'll talk about psilocybin and psychedelic drugs. All right? Cheers! So, is this what you have been waiting for? I tried to tell you a little bit about the brain so that I can try and tell you a little about how these drugs are working in the brain, because I think that's really important. Not just talking about psychedelic drugs and ecstasy for the sake of it, but trying to tell you a little bit about how the brain works and how these drugs works. So when your brain gets stuck, maybe you get older like me, and your brain doesn't become as good as learning or de-learning stuff. So when you're young, you can learn a language in a week. My kid moved from the US to Denmark and learned to speak Danish in 2 weeks from American. Oh, he had Danish parents, but he was only talking English when he lived in the US. And stuff like that, the mind of a child or the mind of most of you people here today are really what we call plastic. And that means that you're really good at changing the connections in the brain. But as you get older, this starts to slow down a bit. So then things can get really stuck in your head. And we're going to talk about how 2 different types of drugs work and how they're used in what we call psychedelic assisted therapy. And assisted is really important because it's not just taking psychedelic drugs, it's taking psychedelic drugs and getting therapy, because we need to do these things with the latter. We need to go up and meet our fear and get the fear response to go down in order to remove the connections that are made in the brain. But some of these drugs can help in 2 different ways. So 2 of the drugs I'm going to talk about are the psychedelic drugs. That's what you call magic mushrooms or psilocybin and or LSD. And they work by increasing the plasticity. So they make your brain young again, sort of. It makes it easier for the connections to be made, but also easier for the connections to be removed. Then we have MDMA, also known as ecstasy. If you have a dirty tablet, that was part of the whole recreational drug use scene. But MDMA is the word we use in science. It's a strong releaser of the compound called serotonin. And serotonin is a neurotransmitter in the brain that is also modulating the connections between nerve cells. It also releases another hormone that's called oxytocin. And oxytocin is what's released in the female when they breastfeed their kids, or when you fall in love and you feel this huge love come over you when you meet somebody. So this is really, oxytocin is really the love drug, so to say. But those 2 together really lowers fear. So it acutely lowers the fear response. And that makes it easier to meet a really hard anxiety or fear and go and talk to somebody about it and then get back again. And without having this fear response going through the roof. So that makes it possible to do the therapy. And it's done in 2 different ways. So psychedelics, if you take psychedelics, if some of you have tried psychedelics, you also know that it's probably not the best time to try and do therapy. So you take a psychedelic and then the therapy is done afterwards. So in the days after. It makes the mind go plastic, and then the days after you can work with restoring or making new connections. MDMA is different. It's not a psychedelic drug per se. It does induce mind-altering experiences. You don't get hallucinations, you don't blend senses, and you don't get that ego dissolution or out-of-body experience. But you do feel a lot of love, and you have the possibility to do therapy while you are under the effects of ecstasy or MDMA. So that's the difference. This is done on drug, this is done after drug. Yeah?
I know that you said you're focusing on the game anxieties and whatnot, but does this have a different effect on neurodivergent brains as opposed to just neuroregular ones? Like for the people with ADHD and all of those?
Yes.
Does it have a different effect?
That is something that is a really good question, and we simply don't know that yet. There is a lot of things that you may want to try out in the future, and you have to do big clinical studies in order to figure out, could you do something with ADHD, could you do something in autism and things like this, if they also have anxiety, and how is it working? We simply don't know yet. But as the whole scientific area about this grows, we will also start to know a lot about these other comorbidity disorders and stuff like that. One part that's out for now. It's the schizophrenic whole spectrum of disorders. And these disorders, they're kept out of all the clinical trials at the moment because of fear of making it worse, simply. So that will probably be the last part that is actually included in some of that. But I'll show you a little bit about the clinical trials going on in just a second. So right now, if you look into the scientific literature and look at how many scientific works are actually being published about these drugs, you can see that up through the 2000s to 2010, not much was done. At this time, it was still pretty much illegal to do research on the drugs in the US, because a guy called Nixon really enforced a really hard law on psychedelic drugs back in the 70s. But then after 2010, it started to getting interesting and people starting to actually publish scientific papers on these kinds of things. So this is the number of papers that are published where you have the words psilocybin, LSD or MDMA, and anxiety or PTSD. So you have 2013 scientific papers currently out with MDMA and anxiety. There's also a lot of clinical trials going on. Most of these trials are with MDMA and PTSD disorder, because in the US there's a big market for war veterans that has PTSD, and they are putting a lot of money into this treatment. So that's why it's really booming up. PTSD is also an easier kind of disorder to frame when you want to do a clinical trial. So if you have a war veteran, you know where the trauma is and you know what the stimuli is that triggers the trauma. So it's a really easy way to define the anxiety, right? So that's why it's an easy start. But there's also a lot of other studies going on just in cancer patients and anxiety and depression and Parkinson's disease, and these things that are connected with the end of life. So most of the psilocybin anxiety papers are on what we call end of life anxiety, so the fear of death. And this is if you're in have a cancer and your treatment is, you're run out of treatment options and you know you're going to die. You can imagine that you will have anxiety of dying, you will have anxiety of a lot of things at this point. And many of the studies here with psilocybin is on these anxiety disorders related to death. That is actually legal in some places. It was the first place it was legal to do this kind of treatment was in Canada. So they opened up for the treatment with psilocybin and end-of-life anxiety a couple of years ago, actually, I think. And now MDMA and psilocybin are put on what is called breakthrough therapy approval in the US. And everyone in here knows what a breakthrough therapy is, because that's the same thing as the corona vaccine got. As soon as you get out and you see positive results in what they call a phase 3 trial, that's when you test it out in real people. If you see positive results, then it will go out and it will be approved. That's the same thing with these two drugs in the US. And that means that they will be approved because phase 3 studies are ongoing and they show good data. So it is going to happen in the US soon. Let's talk a little bit about how they work in two different, inside this fear circuit and storing our neurons sitting in the prefrontal cortex. So both of these drugs have actually been tested in the fear conditioning experiment in mice that were cued with the foot shock and with the sound. And when you give them drugs and play the sound, they de-learn the connection between these two things faster than if they don't get the drug. So you see that if you don't give MDMA or psilocybin to these mice, it takes a long time for them to de-learn that the sound is connected to the foot shock. But if you give them drugs, it goes faster. And the neurons in the prefrontal cortex start to disengage, they start to lose the connections between them, so that the memory disappears faster on the drugs than on drugs. This is kind of like a therapy for mice and rats, because you are exposing them to a stimuli that's not frightening, so you can expose them to something and you can calm them down and tell them and it's not dangerous. So that means that you have to do this. In order for it to work, it has to be together with the therapy. So I'll show you some of the clinical trials that have been taking place. This is the first one. This is with psilocybin, and this is in end-of-life anxiety cancer patients. And this is the first long-term follow-up that was done. It's done in patients that have both depression and anxiety. And when you measure anxiety and depression in patients, you do it with a psychology score. You ask them questions. And I have some of my psychology students sitting here. They can maybe answer this better than me. But you will have, here you will do it with a scale that's called the HADS scale. And that just tells you about how much anxiety and how much depression they have. At the beginning, they have a lot. Not to go into the numbers, but you can all see it's high. Then they get two administrations of the drug together with therapy. And you see that immediately it goes down to very low levels. And they get calm and get rid of most of the anxiety actually. And this actually continues, so this stays on for the rest of their life. So this means that in 1 or 2 sessions, 1 or 2 times they get the drug and then they get a lot of therapy, they basically get cured of their end-of-life anxiety. Right? This goes up a bit because some of the patients obviously die during the course, but it is a follow-up study of 4.5 years. So similar, this is the first phase 3 study in PTSD patients, war veterans in the US that have been out and come back and has PTSD. And you see that the blue line is therapy. Here it's done MDMA together with therapy, and you see that alone works. So here it's called a CAPS-5 measurement of total severity, but you can see that the blue line, it drops with therapy. So therapy definitely helps. It does help these people, but when they get MDMA together with the therapy, it helps even more. So they get even better, and by the third session, they get significantly better than just on therapy. One thing you have to remember when you look at these studies is that they're pretty biased because these patients that are given MDMA definitely feel that they get MDMA. And if you feel you get the drug and you're told that it works, then you are more likely to believe that it actually works. So the people that get the MDMA with the therapy also get a feeling in themselves that this should work, and if it doesn't work, it should work. So it works better. But the other people that don't get MDMA, they don't feel the same, and they say, I didn't get the drug, so maybe it doesn't work. And then that actually exaggerates the difference between the 2 lines. That's really important to remember, that line is actually bigger than it should be, just because of the placebo effect.
But normally a placebo is like you think you took the drug.
Yes.
You should think that you had it.
But in these cases, nobody thinks they took the drug. Everybody knows that they get the drug if they get the drug. It's no hiding it, unfortunately.
May I also just ask?
Yeah, but I'm also going to do a lot of questions in a minute, but you can also ask me now. I can't see who you are because you're sitting behind the light, I think.
Sorry. I was just curious about the come down effect after the prescription of the MDMA, for example. Does the come down effect, the low after the high, also make a difference with treating PTSD in war veterans?
They in London or in the UK, they did a study on the calm down called Blue Monday, and they actually didn't see any Blue Mondays in the people that were treated with therapy and MDMA in the sessions. So nobody reported any Blue Mondays actually.
Is the placebo effect that they think that they're taking MDMA? So the placebo drug that they're taking, do they think that they're taking MDMA?
No, in these cases they were given MDMA and therapy, and then they were looking in the day after if they were feeling worse on the day after, and they were generally not. So I don't think that feeling of feeling worse the day after is from taking one dose of MDMA. It's a combination of everything you do when you also take MDMA. Huh?
If you're taking a placebo and you're taking MDMA, maybe you have counteractive. Like if you think you're taking MDMA and then you realize you're not taking MDMA, you might have negative effects.
Yeah, that's what I tried to explain before with the line here, that it was actually
So then there could be another one there, which is like a placebo of another type that's between that, without thinking that.
the true difference between these lines will be smaller, right? Because of those effects. So also, I like to show this one. Also, there's been 3 different studies out on how dangerous are these drugs if they in the recreative environment. And they look at 2 things. So they look at how dangerous are these drugs to others and how dangerous are they to the user. And there's been done a study in the UK, in the US, and in Australia, and they all show pretty much the same thing. Alcohol is the #1 dangerous drug when you look at all these drugs together. Heroin, crack cocaine, methamphetamine, but you can see that down in the other end of the scale, you actually have magic mushrooms is psilocybin, LSD, MDMA here. That doesn't mean that they're completely safe, but what I do want to show you is that another drug that you use to treat anxiety is placed here, that's benzodiazepines. So they are all considered safer than benzodiazepines. Right? So I really want you to remember these things when you leave today, that anxiety has a clear neurobiological origin, and you can learn fear, but you can also unlearn it. But it takes a lot of psychological work. There is help to get, first from a psychiatrist or psychologist, and second, if you're old enough as me and stuck and your brain is not plastic anymore, then possibly with medication as well. And that's it for me for now, but I would like to answer all your questions.